Welcome back to Birds of a Feather Talk Together. This week we talked to Kevin Burns about tanagers. Kevin, an ornithologist and professor of biology at San Diego State University, goes way back with Shannon and John and led the research that reclassified what exactly a tanager is and what it isn't. Many birds which have the common name tanager, such as the scarlet tanager or western tanager, aren't actually tanagers, and birds like Darwin's finches actually are tanagers. Many of you might already know this, but we get to talk to Kevin about what exactly went into this research and what that process was like. We also hear some fun stories about Kevin, John, and Shannon in the field studying birds and talk about how impressive many of the different tanagers are. It's a lot of fun, and I think you're going to enjoy it. Okay, go grab your binoculars and let's get into it. Kevin, we're really excited to have you on. Shannon's been trying to get you on for a long time, and I'm glad this has worked out. Shannon, do you want to give a little intro to Kevin?
I've known Kevin for almost as long as I've been in science. So I met you probably in 87. Is that right, Kevin? That makes us,
Yeah, it would have been 88 or 87 when I visited for
yeah. So
grad
that makes
school.
me really old, him less old. But
Still old.
so Kevin came to LSU to go to graduate school. And he grew up in Texas. He can tell us a little bit more about that. But now he lives, and he's very soft because he lives in Southern California in the land of no weather, except nice weather. And Kevin,
Would it be 100 degrees today?
Oh, wow.
where? In
Oh.
Tucson or in Phoenix?
No, right here in San Diego.
In San Diego?
Whoa.
Yeah.
Wow, that's pretty hot. And Kevin and I lived together for several years, off and on, with and without John at varying times. And he was also my maid of honor at our wedding, at John and my wedding, which kind of flipped every wedding person in Louisiana out because they couldn't figure out why I didn't need a dress. They kept saying, no, you have to have a dress. I'm like, he's not wearing a dress. Honestly, he's not wearing a dress. I don't need a dress. I need my dress, but not him. So I've known him for a long time. We did a bunch of field work together. Kevin has a plan for everything. He has a contingency plan for every possible thing that could go wrong, including things you've never thought of before. So it's always interesting to go places with Kevin. The only thing...
That way you're never disappointed.
Yeah, well, I was, though, because the only plan of yours I rejected was your idea that a flashlight would save us from a bear coming into our tent. Flashlight under his pillow. Yeah, well, I don't think that had a chance in hell.
Wait, did you guys have a run-in with a bear?
No, but they could have.
Yeah, it's all hypothetical, I think.
Yeah,
Okay.
this is like part of he probably had a plan for malaria, which is not common in the Sierras of California.
So Kevin, I know we're here to talk tanagers. We'd love to hear just from your perspective, kind of, your background, what you want to talk about with tanagers and kind of kick things off. Do you want to give us a little intro?
Sure, yeah. I'm originally from Texas, grew up in Texas and did my undergrad at Texas A&M. And then for master's, I went to LSU, and that's when I met Shannon and John. And actually, I think it was John's roommate first, and then it was Shannon's roommate, and then it was John and Shannon's roommate. So had a lot of good times at LSU, a lot of great memories there with them. And then I went to Berkeley and got a PhD there, and then did a postdoc at the Naturalist Museum in New York City. And then I've been in San Diego, working at San Diego State ever since. So about I'm almost at 27 years here. So, but yeah, tanagers are just, I don't know, they're an amazing group. And it's something I've been working on now really since my PhD. So over 30 years working on them. They're just like, I don't know, they're pretty amazing. So I thought we could talk about those.
Awesome.
And it's a spectacular example, as Kevin will be able to tell us, about what molecular data has taught us about what we didn't know about relationships. So the tanagers that Kevin originally studied actually aren't tanagers anymore.
Wow.
Yeah, that's kind of true. I mean, there's the problem with or what was difficult about tanagers is no morphological character defines them well. And people had talked about this for I don't know, really decades about how difficult they were. And it really wasn't until we had molecular data available to solve that problem. So I first started working on them because they have a lot of even what we thought they looked like at the time is like 240 species. They were pretty variable in color and in morphology. So I thought, okay, here's a chance to study, evolution of feeding adaptations, classic evolutionary subject, or evolution of color diversity. So we could look at sexual selection. But I didn't, we didn't even know what the hell a tanager was. I can say hell, but we just it wasn't until later when we sorted out with all the DNA. I probably would pick a different group if I'd realized kind of what a mess they were. But I'm really glad I stuck with it. And so it's been kind of a journey.
Don't you think your work just highlighted to not just to ornithologists, but to every bird watcher in the world that we don't know anything? We didn't know anything about the tree of life back then, and we shouldn't make assumptions or use common names to decide what something was.
Yeah. I mean, yeah, it's just been so many changes, right? So, and you're right. We're stuck with a lot of language and baggage from before we kind of had the tree of life. But sometimes I reflect on how much those folks got right? Because all they had was maybe specimens, because not all the people that were classifying were actually in the field. So the specimens and maybe distribution, they were trying to sort all this stuff out. So there's a fair number of tanagers that got right, but a lot of them were not really in the right place. So we went from 242 species when I started my working on it. That was sort of the main classification used. And now we're at 385.
Oh, wow.
But there's even more shuffling than that because we lost a lot of birds. They went to other families. And then a lot of other families came into the tanagers as part of this process.
So my favorite example with respect to that is the tanagers, the quote unquote tanagers in the common name sense that people put in the genus Paranga, that are the ones that people in North America will actually know.
Summer
includes
tanager,
those
scarlet tanager, western tanager, for example.
Hepatic tanager, those aren't tanagers. So I have to tell people if those are the only tanagers they've seen, they haven't seen a tanager.
Oh,
Oh,
that's
no.
right. That knocks us out.
Yeah.
No, and as I was preparing for this, it really hadn't dawned on me from a biogeographic sense that the only members of Thraupidae, that the true tanagers in the U.S. are actually white-collared seed eater, which gets into Mexico, and bananaquit, which occasionally shows up in Florida, and that's pretty much it.
Well, there are a few others that pop up in Florida as vagrants, but you're right. That's about it. There's a seed eater that gets into Texas, and then some random things in Florida. I guess one of the honey creepers has been showing up lately in Florida and even in Louisiana. And those are thought to be natural vagrants, not escapees. And in San Diego, we have one of the seed eaters that's a escapee from the captive bird trade that you can see occasionally. But probably the best place to see a tanager in the U.S. is your local zoo, because they're kept in captivity a lot. So you can see them there. And in San Diego, they keep them at that so-called hummingbird aviary, which only usually has like one or two hummingbirds in it, but has lots of tanagers.
We'll get into some of the things that Kevin has found, but when I was preparing for this, and I do this periodically, like maybe every year or so, I look online to see what publications say about what a tanager is. And so I looked again this morning, and the only place I saw that didn't include scarlet tanager and things like that, like the Britannia, the encyclopedia people have it wrong, is a site called Kittle.com. And they had all of the modern stuff about tanagers. So the kids' encyclopedia has it right. The grown-up encyclopedia, every single version of it is wrong still, after 20 plus years of knowing something pretty fundamental about the relationship. So kids will know better what a tanager is than grown-ups who look at the encyclopedia. And even then people who look at their textbooks, where it's mostly wrong, even today.
But to kind of get back to something John was saying earlier, yeah, when I originally started my PhD, I was just going to look at Paranga, the scarlet summer and hepatic tanager. And so I had to root that tree and put in the other act groups. And so that's how I started putting in tanagers into the tree. And then just weird stuff started happening from there. And so even though I thought I was studying tanagers at the time, I wasn't. But then as I expanded outwards, then things started making sense.
And also, I think one of the interesting messages in all this is the data that you originally started with morphed tremendously as we began to get more and more capable of getting genomic type data.
Oh yeah. I probably spent 2 or maybe 2.5 years of my PhD sequencing like a few hundred base pairs of every species and feeling amazing about it. And then we got the automated sequencer my last year, and I just kind of almost threw all that data out and started over, and then added all these other species I never thought I'd be able to add. And everything was a lot quicker. And now, I mean, it's routine to get genomes of things. So things really just have accelerated.
And it was amazing that so much material was available in tissue collections that you guys were able to use too.
Yeah, I mean, LSU's was prominent in that because they had done so many years of collecting in the neotropics and saving stuff all the way back, I think, to the 70s or something. So, and it was fun to kind of, when I was at LSU, a lot of people were working on neotropical birds, but I wasn't. I was working on fox sparrows, which I loved my project, but I was also jealous of all the neotropical stuff people were doing. So when I was able to start on that in my PhD and then go back to LSU and kind of look at the specimens and use the tissues and things like that, it was kind of exciting.
Kevin, was the first time you went to the neotropics when we went to Costa Rica? When was the first time you went?
Yeah. So, well, the first time, I guess the first time I actually went to the neotropics, I did a trip to Mexico, but I didn't, at the time I saw a saltator, which wasn't classified with tanagers. So at the time, but the first time I actually saw a tanager would have been on that trip I went with you guys to Costa Rica. And I think we were stuck in Costa Rica, as you remember, because things were closed when we first got there. And I think, Shannon, you twisted your ankle or something, or you were sick. And John and I went to the zoo, and we saw palm tanagers there, and I think blue-gray tanagers. And so that would be my first tanagers.
In the Costa Rican zoo?
Yeah, outside the zoo. Sorry, the zoo is closed. But then, of course, we saw lots of tanagers on that trip, including you guys already did you did a podcast earlier on the Acanthodops, pig-billed finch, which is a tanager. Yeah.
So that's my first time seeing a tanager then too.
Yeah. Yeah.
Wait, peg-billed finch is a tanager?
Yeah.
Yeah.
Oh.
Tanager.
I'm so confused now.
Me too. Yeah.
I wonder, is that that might be my actual for real first tanager. Because it's the first bird I saw when I went out of La Jorgina across the road into the...
Right. Because we did try to go birding that one day, like Christmas Day, I think it was. We took a taxi out to Barulio Carrillo, but it was pouring rain, and we didn't see any tanagers on that trip.
Well, we also, you guys are forgetting, because remember, the offices were closed because it was
Christmas.
New Year's.
Yeah.
And so we borrowed that car, and we drove all the way down the coast. And so we would have seen some tanagers on that.
But we stopped at La Jorgina first. So,
did we?
Yeah.
I've forgotten that.
I think Shannon's right. We stopped at La Jorgina on the way down to the coast. So, yeah. But the tanagers that stick out for my mind the most from that trip are the scarlet rum tanagers, because they're just so charismatic and colorful and active. These are dark birds with bright red rumps, and they were pretty fun. And then Shannon was studying those at the time too.
Yeah, well, they have an unfortunate habit of being near people, which makes them kind of actually hard to study. So I thought I was going to be the only person ever to want to capture one of the most common birds and be unable to, because you had to go into people's gardens to capture them or things like that. You had to get permission to go in their houses. It was very odd.
RJ, you were mentioning peg-billed finches and surprised that it was in the Thraupidae.
Mhm.
And that was, that's a perfect example of one of the taxa that had been placed in another family. It was with the warblers, wasn't it, Kevin?
I think I think it was with the sparrows. I think
Yeah,
that one was.
I think
Yeah,
Okay.
that's right.
yeah, yeah. The Coney rostrums, the codebills, were with warblers,
Mm,
but now they're in my group. They're in the tanagers.
crazy.
So can you explain the process a little bit then of just like researching a bird, determining that it's not a tanager, it's something else, or that something is a tanager, like just kind of, I don't know how you determine that.
Well, like if there was some sort of morphological character, you could look for that morphological character. In this case, there wasn't. So you had to just kind of go through and do a bunch of DNA sequencing of different birds and try to figure it out. And I started by doing just kind of a one of each genus originally, and so that helps kind of sort things out. And then
And you need a
expanding that.
tissue sample in order to do that, or?
Yeah, sorry, you need tissue or blood, or back then when I started, it was really hard to get it out of like toe pad or skin or things like that. So I was mainly relying on museum collections and that sort of thing. But yeah, so every time you get a sequence, it was almost like I couldn't wait to see where it was going to fit. And so it was like a little game. I'd go and put it in a quick analysis, not a good analysis, but a quick analysis and see, oh, okay, it's sitting there. Or what's it doing over there? And just had to keep adding more and more. Finally, I started collaborating with other people working on warblers, blackbirds, sparrows, cardinals. And it wasn't until we put all that together, like 800 something plus species into this giant evolutionary tree, was the data able to sort out and show us where the groups are defined by the DNA. And that's finally when I felt like I had a good sense of what a tanager is. And there's still been a few surprises since then, but pretty much since we published that paper in I guess it was like 2012 or 2014, around then, I think we have a pretty good sense of what it was.
And this happens across the avian tree of life in various areas as people get more and more molecular data. Is when you combine these data sets, you start getting things you didn't expect. And I mean, again, the first time, if you only have one individual of one taxon and it comes out in a weird place, the first thing you wonder is, what did I screw up in the lab?
Yeah, which happens.
Well, because sometimes the things Kevin found were so shocking to people that would have been their first thing, is that he just messed up, labeled a tube wrong, labeled a line wrong in the data matrix, because he can't possibly, I mean, we heard this a lot, it can't possibly be like that. And
Yeah, that's true. Like there was a whole set of birds in tanagers called euphonias that turn out to be goldfinches, which are they're fairly close related, but not really. They're kind of outside that whole New World 9 primary group. So there's, hundreds of species closer to tanagers than euphonias. Euphonias are more closely related to house finch, for example. And I would like to say, though, before I started my work, we did already have a hint or know that some of the finches and sparrows probably went with the tanagers, because there was early work done by Tony Bledsoe, and he published a paper that with, I can't remember, not very many species, but some of those sparrowy, finchy things were coming out with the tanagers. And then Charles Sibley did some work on this too with an earlier DNA techniques. So I did have some idea that there was stuff, all these finches were going to go in, but I just didn't know how many, and then how many things were going to lose from tanagers and all the other things that were going to come into tanagers.
so, Kevin, what would you say is the most, besides the tree, like now you have the tree in there, you can look at other aspects of their biology and evolution, distributions, etc. For you, what was the most influential thing that tree contributed to?
Well, I don't know if I'm going to answer the question with what you told me not to answer it with, but it sort of changed the whole perspective of the group, because before, the idea of tanagers were they were very colorful birds, and they weren't good singers and all these things. But now with this new group, like 385 species, they have really all different kinds of colors, but they also have a lot of drab colored birds in the group, and they have a lot of essentially different birds. Males and females look different, at least from a human visual perspective. It's a whole other story when we talk about the way birds see them. And we can get into that if you want. But then, and they're found in all the habitats pretty much in terrestrial habitats in the neotropics. There's even one that nests on glacial ice,
wow.
and then they're in the rainforest and they're high up in the Andes. They're just everywhere. And so that sort of changed the whole perspective. And then they're removed from the United States and Canada, right? So they're just this neotropical group. But there are some on in the middle of the South Atlantic. There's a group that got out to Inaccessible Island, Go Island, Tristan da Cunha, just one of the most isolated places on Earth. So there are some tanagers out there. So it's pretty crazy.
Yeah, I get to see one of those in the Falklands, or at least it's actually part of the same
relative.
species as the one
Yeah.
in Argentina, but
Yeah.
it's pretty clear that the one on Inaccessible Island is a derivative of that, in my opinion.
Yeah.
Looking
exactly.
morphologically.
Yeah, it is. Yeah.
I mean, I use tanagers all the time when I talk to people about our collections and about evolution. So I always you're a part of every tour I give. And it's because for me the kind of a foundational thing that came out of your study is that it just was a very strong reminder that we study plumages from a male perspective. Ornithologists have for centuries. That was the focus. But the tree of life that Kevin found showed, and his publication showed this very clearly, that if you want to know what's happening, you better look at female plumages, the evolution of drab and brilliant colors in female plumages, and you cannot get even close to the entire picture of plumage evolution in birds if you don't study females too. They're not just like drab males.
Yeah, there's lots of tanagers where both males and females are colorful. And so that's somewhat unusual in the avian world. And so female plumage overall changes more across the group than males. And so it can't just all be about sexual selection acting on males. There's definitely things going on with females.
So you'd never look at those brightly colored birds a priori and think, oh, the actual changes are in females, right? Because you're just blinded by the magnificence of the male plumages and all of the colors and that tanagers have explored through evolutionary time.
Yeah, some examples of this are birds like paradise tanager, which is this Amazonian. It's found all the way across the Amazon basin. Both sexes are the same, and if you look it up, in Brazil they call it sete cores, which is 7 colors, and it's just got some of the most vibrant coloration. But males and females are inseparable in the field.
Yeah, it's got this. It's my favorite tanager, so I'll talk about it for a little bit. It's got this apple green head, mostly black body, bright red rump, like a violet, purple throat, turquoise belly. It even has ultraviolet coloration embedded within that color as well. So it's just got about everything. And then we've measured all the tanagers and quantified their color. This is what we do in science. We take all this beautiful nature and turn it into numbers. But when we did that, we could then rank the birds in terms of most color complexity, and the paradise tanager wins, just in terms of all the different areas of color space that it occupies.
So one early hypothesis about paradise tanagers was that they might be poisonous, and maybe that was actually a posomatic coloration. And I can still remember a colleague of ours, John O'Neill, who was at LSU, when we were talking about it, when he was with us in Costa Rica, and he just said, I've eaten them.
Was he serious?
Yeah,
he was.
Whoa.
He just tried it at some point because he wanted to know if they were actually
poisonous. He tried it
poisonous.
because he tried everything.
Well, that's true
He
too.
tried every bird that they captured and made into specimens. He ate a piece of every bird, and that for him, that's a lot of birds because he was one of the first people to do wide scale, kind of more modern collecting in the New World tropics. That was like a philosophy he had was to eat them. So they cooked them. I'm not eating them raw, but yeah.
Are there birds that are poisonous to eat?
Yes.
There are.
Really?
And we can do a podcast on that with the curator of birds at the California Academy, who's the one who discovered that. He was a University of Chicago graduate student at the time.
Oh, wow.
Yeah, for another day.
Yeah, definitely want to talk about that.
But I think, despite all their bright colors, none of the tanagers are known to be poisonous in any way. And there's a tremendous variety of color in them. So what are they doing?
Yeah, you wouldn't. I mean, why would males and females of paradise tanager be so similar? I mean, what is being signaled by those colors?
I mean, they both could be territorial, right? Some of these birds, both males and females, in colorful. There's no reason why females wouldn't defend territories too sometimes.
But you don't need to look like that.
That's what I was going to say. Does territory defense have to do with coloration? Is it like
Um,
hairy or?
I don't know. I feel like it might. Now that you say that, maybe I should backtrack a little bit. I'm not sure if that there's a lot of evidence for that. I mean, I guess maybe there could be male choice on female color as well. I don't know. Just speculating here.
I think one of the real things it illustrates is just that there's a lot more we still have to learn. So as cool as paradise tanagers are, and you can see them, and they'll come down if for instance they'll come down to feeders in camps in South America, in the Amazonia, up in the trees. They go around in these mixed species, or these species flocks, monospecific species flocks, where you can get 5 to 10 together. And so I don't even know what the makeup of those flocks are in terms of what the individual status are within the species. And I don't think anybody has ever been able to catch a whole flock so that they could figure out how they're related to one another, for instance. So there's a lot we don't know about some of those details.
So, of course, I've only seen them dead, not alive, because that's the way I've seen almost all birds, dead, not alive. Um, are there elements of their behaviors where they flash some of those colors or hide some of them? Because those are, Amanda, that's another way in which birds can use color, right? They can flash it as a signal of territory defense or whatever, like red-winged blackbirds do when they're breeding. But do they do that?
I mean, I think they highlight them in courtship, but I don't know about flashing them against predators and things like that. And a lot of the really brightly colored tanagers have a lot of different patterns on their face, which makes me think that that's also involved in courtship. They're, involved in birds that are looking at each other, not something coming from far away.
Oh, that's cool.
And I do want to make a point. We keep saying that they look the same, the male and female paradise tanager, but we can't tell the difference, but the birds can. And this is where studies where we've used, especially one of my former grad students, Alison Schultz, we looked at, we quantified the color and then we put it into a model of avian vision. And we found out that pretty much like 98% of tanagers are sexually dichromatic from an avian vision. From human vision, it's about 50%, but 50% dichromatic, sorry. And then from avian vision, it's 98%. Yeah.
Oh, wow.
So almost all of them look different to other birds. So they can tell each other apart by sex.
Kevin, is that some of that ultraviolet?
Yeah, it's ultraviolet. They have ultraviolet. They can see into the UV spectrum, so a lot of the colors reflect into the UV spectrum, and that we can't see that. And then they also have better abilities to discriminate among colors than we do, due to the way that their retinas are structured. That they can see different shades of color better than we can.
And I just, one of the challenges we have here is that, as Kevin was saying, now there are how many species?
385.
385. So, we talk about paradise tanager, and that's 1 out of 385. And an awful lot of them don't look at all like paradise tanagers and don't behave like paradise tanagers, and don't occur in places that are like where paradise tanagers live. So it's just an incredibly successful radiation.
They're all different diets: nectar feeding, insect feeding, seed eating, everything in between. They're in all those different niches, all the different habitats. It's just really everywhere. It's the second largest. It's the largest songbird family, and it's the second largest family of birds. Flycatchers are the only ones that beat them out, and it's really, I don't know if I think it's pretty much the largest radiation of kind of exclusively neotropical birds.
Yeah, I was because that influences our understanding of the biogeography of songbirds. And that was completely transformed by this tree and the knowledge of what does and doesn't go inside of this radiation. And it was that what John said is true for me. I had never really thought through before that just how rooted in the neotropics tanagers were compared to what I used to think about them. And then we're going to talk about some more of these birds, but people should know that things like Darwin's finches aren't finches. Darwin's finches are tanagers. Yeah, that was another really
That
fun.
was one of the biggest surprises,
Hmm.
that finding.
So will the common names ever change? Like we're calling things tanagers that aren't tanagers, we're calling things finches that aren't finches. I mean, is that ever gonna
I
change, or is that
think possibly, but it's such a long road because there's so many birds that would fit under. It would be easy maybe to do some of the ones that we have in the US, like the Paranga tanagers. And there has been talk about changing those just to Paranga because it's a pretty easy word to say. And I guess Paranga is one of the native language words for tanagers. And so maybe that makes sense, but maybe it doesn't since it goes with tanagers and they're not tanagers. But at least we're not calling them tanagers in English anymore, I suppose. But yeah, I think it's hard because, we'd have to change so many names. And, we have a lot of issues about changing names, right? So maybe if we don't have to change them all. But it is frustrating to kind of have to, and people get frustrated too when they find out, well, those aren't tanagers. And I thought they were tanagers. And you have to explain it to them. But there's lots
Yeah.
of other cases of birds that have the wrong last name, if you will.
I was
Although
wondering oh, sorry, Kevin. I wanted to ask, like, for you guys, for ornithologists, do you even care about the common names? Like, do those matter at all to you?
I
So,
do.
so, so Kevin, you want to wear your hat of the AOS checklist committee?
Well, I mean, I care about them because as a birder, I like to use the common names. And I do sometimes, especially when I'm talking about tanagers, I forget the common names and I just use the scientific names, but that doesn't help when you want to talk to birders and stuff. So I don't know. I think the common names are important. It's important to have a common language, and it's important to be thoughtful on what those common names are, I think.
That works better for North American birds' common names. They kind of break down when you get outside of North America or Europe, because
Right.
there's not common names. So every place
Yeah,
can have a different name for that bird.
Yeah, we should say these are more like English names that we've come up with rather than common names. It's the standardized list that the AOS comes up with that eBird mostly adopts, but not always. eBird's kind of doing some things differently sometimes.
And Amanda, one of my formative experiences in the neotropics was going down and becoming a bird bander down in Manaus, Brazil years ago. And I ended up in the field and none of the guys working for the project, the Brazilians that were working for the project, knew any of those English common names. They used all scientific names. And so a lot of times for us, the scientific names work better, but again, one of the most amazing things again about the stuff that Kevin's done with this group is they've realized that a lot of the relationships were problematic with the traditional binomial system of nomenclature that we had. So the genus now didn't represent exactly what the historical composition would be. And what do you do with that?
Yeah, pretty much any genus that had more than 5 species in it turned out not to be like a clear group, a genetic group. We had to come up with all these different genus names and resurrect old ones. But so we really shook up the taxonomy. And I'm confused sometimes as well because the names so much, but you have to do that so that you can reflect the tree. So there's the genus. I can't remember how many genera names we changed. We had to do that because any of the bigger groups, like there was one genus Tanagers with, I think it had 50 species in it, but then there was a whole other genus Thraupus embedded within it, which was kind of a surprise, but then we had to kind of accommodate that change.
So you're personally responsible for the confusion that many bird watchers feel about these things, and probably
Oh,
the the anger at change that can come out when you find something new like that.
yeah, well, I'm angry too sometimes, but but and the warblers too. When we work on warblers, that's where people hear about more. But I should say I want to make a point about tanagers that we haven't talked about yet. That the rate of speciation in this group has been off the charts. Even though it has 380 something species, the group is only 12 million years old, which is not very old for an avian family, and certainly for that many species. So the rate of speciation is something like 40% higher than its close relatives, or an order of magnitude than vertebrates as a whole. So that's just kind of explains why sometimes it's been hard to sort them out because of all that radiation. But then it makes it even more remarkable that they're able to evolve all these different beak shapes and colors in just that short amount of time.
So that's where the tree leads me to the next things, is that once you have that tree, you understand the context from which you can learn about the evolution of beaks. I don't think there's a group of birds on Earth that has more beak morphology variation than tanagers do, especially once you stick into the tanagers all the things that didn't used to be in tanagers, which expanded dramatically the amount of beak morphology variation. So what is it about tanagers that makes them do that? We don't know. Other groups of birds that are 12 million years old don't do that. They all look, like fairly cookie cutter size differences along the same theme. Um, but there are even groups of tanagers that even go outside of what the average tanager is like too. They take it to even bigger levels um, than before. Darwin's finches are one of those. So they're doing all kinds of, they're exploring all kinds of morphological space that other groups of animals don't, and we don't know why. And the tree is the fundamental thing that tells us, that will help us understand why. You cannot study beak morph. I'm getting on my soapbox. That tree is so fundamental for understanding that you need to study these things in a much broader context than they often are studied in. So you don't study beak morphology variation only in Darwin's finches or only in Hawaiian honey creepers, because there's so much more to that story than just that.
Well, and this may be arrogant on my part, but having grown up learning some of the things Kevin was talking about earlier about what people had discovered in the past using various what they had available to them, like morphology. And I love the fact that he points out that a lot of times they got a lot right, but then I guess the one thing I'm convinced of, and time may prove me wrong, but is that the data that we have now has actually put the tree in a position that it's highly unlikely that we're going to get some other data that completely upend what we're thinking now. And you always want to knock on wood when you say that, because who knows? But at the same time, I'm convinced that even though colleagues of ours at the American Museum, when we were postdocs there, used to say you can never know the true tree, which is true, right? We don't know. We don't have a time machine to go back in time. The data that we've got are incredibly convincing that these relationships are actually going to hold true and down the road.
Yeah, it's exciting time to kind of be in this field. And John and Shannon and I kind of traveled that journey from where really all you had were morphology, then to look at proteins, then DNA, now whole genomes. And Darwin writes about having a dream of having a whole tree of life of everything, and we're going to see that fairly soon. Well, that's another subject, but we don't know all the species that are out there, right? But for the species that we do now, we'll probably have a pretty good tree, and not too long from now. And certainly groups like birds that are known pretty well, we're going to have a good tree, maybe even within a decade for everything. What do you guys think?
Well, I'm convinced that I have to learn all these new genera that you've made.
Flashcards.
Oh my gosh.
I'm true about the flashcards.
I don't think that, Kevin.
Oh, really?
No?
First off, I don't think we have any idea what a species really is, but that's my thing. It's not the first time you've heard me say that.
Well, what about birds? Like with birds?
We don't know what a species of many
Yeah,
birds
but
are.
okay.
No way.
Yeah.
Maybe we will have it, it just gets refined more and more. I think it's maybe arrogant to think that we're that close to that.
I think it's really important to detail what the question is. So I think the basal relationships of birds down to the genus level in a lot of situations are actually going to become pretty stable based
Nope.
on the data that have been available.
Nope.
All right.
Not at the base, because the base is the worst part of avian.
Well, all
ways of
right.
life.
So
Or
yeah.
it'll be unknown. Like there's no more data we can get to
That-
sort of
well,
help with that
That could be.
until we invent John's time machine.
Well, I just laughed because again, for Amanda and RJ, one of the things that we've struggled with over time is even as we get these different types of genetic data, is some parts of genetic data are better at analyzing things than others. And so there is an aspect of picking the sets of characters, even within molecular data, that you're using to come up with your phylogenetic hypothesis that you need to think about. And so I guess, maybe I'm jumping the gun and saying that I think there's a lot of stability in all this, but it'll be interesting to see.
The reason I say that, Kevin, is that we haven't come close to bacteria. That's why I think that we're really not close to it. And there's way more species of bacteria than there are of anything else, or species, whatever that is in those things. And what do you consider viruses? Are they living or not? There's a lot we don't know about the tree of life for viruses as well. So that's why I say we won't know it for 10 years, because those things are really
We
common.
probably only know 10% of the species that are out there or less. So,
Well, I think we're kind of getting close on time.
Do we have a mailbag question?
Oh, we do. Yeah.
So, Kevin, people send us questions pretty frequently that they want answered. Some of them are a lot harder than others.
So this is from Kathy from Skokie, who's written to us before. And Kathy said, so much is made of the spring migration from south to north. Followers put out food and gather to observe, but what about migration north to south? Do the birds take the same route, a perfect round trip? When does it start and end? Shouldn't the night lights be turned off again?
So thanks for that question. So the quick answer is yes. And fall migration in a lot of ways actually involves more individuals than spring migration does. And the reason for that is the birds have gone up to their breeding areas in northern latitudes and produced a whole bunch of young, which have to migrate through. So we're right now in the midst of fall migration through the Chicago area, and there are going to be millions of birds coming through in the next little while. And so turning off their lights and closing curtains and things is really important. And, nothing has illustrated that more than actually a fall migration event last October, where 1,000 birds hit McCormick Place on a single night. And, this is one of these things where we have the tools to study migration better than we've ever had before. And so we can do a lot to mitigate some of that. And one of the good things about that event is that's opened people's eyes in a lot of ways. McCormick Place has spent $1 million putting up decals on the windows. And we're going to hope that actually reduces a lot, some of the mortality that happens there. And the other thing is we're going to keep monitoring it. So yeah, it's definitely, fall migration is definitely an important aspect of the whole process that we have with all these long distance migrants.
And bird deaths to try to minimize the number of deaths against buildings is a collaborative process between biologists, people who care about birds, and people who manage and own and design buildings. And as we get more of these relationships, tighter relationships, building design can be done better. Lighting regimes will be better. We know the problems. Now we have to get solutions. And then we need government, because we need governments at all levels to start requiring bird-friendly building design and construction and materials, so that the costs come down. Yeah. But, we actually save these birds so that people can learn all kinds of things at the Field Museum with these collections that nobody else in the world can do. So our collections have this unique ability to tell us a lot about migrating birds. But we know very little about the annual cycle of most migratory birds. We know something on the breeding season. We might know something on the wintering season. We don't know very much about what happens in between unless they hit a building.
And one thing we know, thanks to Kevin, is there are no throupids in those data sets.
All right. Well, I think that's a good place to call it then. John, do you want to close it out?
Yep. Kevin has done an incredible amount of work, and he and his students have had a lot of fun doing it. And that's one of the greatest things about the kind of science that we can do these days.
All right. Well, thanks, Kevin, for joining us. We appreciate it. We'll
Thank you
talk
guys
again
very
soon.
much for having me. I enjoyed it. It was a lot of fun. And I appreciate John mentioning students, because students have been a big part of all of this work as well. And I feel like I haven't mentioned the dozens of students that have been involved in this project over the last several years. Thank you.
Thanks, everyone, for joining us. We appreciate it. If you have a question for us, you can reach us at podcast.birdsofafeather@gmail.com. If you've been enjoying our podcast, please leave us a review and rating in your podcast app. This goes a long way with helping us get exposure, so if you don't mind, we'd really appreciate it. And see you all next week. We love you all.